Well, first of all, I don't know if we'll be able to see the slides. The lighting situation isn't the best, but we'll do what we can. I can tell you it may be boring with the slides, but it would certainly be boring without them. I was telling Jonathan, he gave a 2-hour slot this afternoon for this talk, which seems to be kind of a mistake, because I can possibly take that and more, and being someone who's able to ramble on at some length about these things. But hopefully this will be of interest to some of you. Those of you that were at the Seeds of Change conference last year can be excused, because this talk is essentially the same as that I gave at that time, and with a few other elaborations. So if you don't want to sit through it again, I fully understand, and I urge you to leave now while the leaving is good. The name of this talk on the program is Alkaloids and Evolution, which is kind of a truncated real title, because we're going to be talking about a lot more than alkaloids. But Alkaloids and Evolution was easy to get on the limited space. What it really should be called is Plant Allelochemicals and Plant-Human Coevolution. But that seemed like somewhat more of a mouthful. And some of what I'm going to talk about today is kind of more established. There's not a whole lot of controversy. The understanding is maybe limited, but most scientists, people that study these things, are in general agreement. And other aspects that I'm going to be talking about are definitely in the realm of speculation. And I'll try to kind of give you notice when we step off into the hypothetical realm and away from the more established areas, just to let you know, if nothing else, that I sort of have a grip on what the difference is. I guess so. The reason I started out with this illustration, which is from the book Parallel Botany, if any of you know that book. It's a charming little book, and it talks about a completely hypothetical, completely fictitious plant kingdom. It's basically a book of anatomy for a bunch of plants that never existed, at least not on this planet. But I put this up here to illustrate the point that even though plants and animals have an extreme relationship of interdependence with each other, plants are not at all like people. And they're not at all really like animals, even though we as part of the animal kingdom and the rest of the biosphere really depends on plants. For one thing, plants by and large don't move around. They are stuck in one place. And so in order to develop evolutionary strategies for protecting themselves, getting along in the world, they have tended to evolve biosynthetic strategies as opposed to behavioral strategies. In fact, one famous botanist, Tony Swain, put it very succinctly. He said, plants have substituted biosynthesis for behavior. And if you think about it, that's an interesting concept. Plants are chemical virtuosos. A greater variety and a greater number of complex organic molecules are found in the plant kingdom than have ever been synthesized by all the organic chemists since the beginning of time. Plants outstrip them in terms of varieties of different types of compounds. And the reason that plants are able to do this, one of the main contributing factors and a kind of neat trick that plants can do that's unique to the plant kingdom is that plants are photosynthetic. I put this up here. This is a rendering of the of the cosmic ayahuasca vine by a Brazilian artist. I put it up here to suggest the idea of photosynthesis. Actually, I guess I could have put an electron micrograph with a chloroplast or something, but this seemed more appropriate. Plants have this ability to take simple elements, carbon dioxide, water, and sunlight, and transform them through the process of photosynthesis into complex organic molecules, the molecules of life, sugars, lipids, amino acids, nucleic acids, all these things basically proceed through the process of photosynthesis. All other organisms in the biosphere are essentially parasites on the plant kingdom. Because, well, not all, I mean, you want to be careful about using those kinds of terms, but most organisms in the biosphere are heterotrophs. That means they don't make their own nutrients. They don't make their own sustenance. Plants are autotrophs. They can, out of sunlight and water and carbon dioxide, manufacture their own food. And that's what the process of photosynthesis is. As energy impinges on the biosphere from outer space, the plants, through chlorophyll, are the first to grab that energy and do something with it. And then from that point, as this electromagnetic energy becomes transformed into chemical energy, it basically cascades through the entire biosphere. Everything else feeds off the plants, but the plants are what is able to fix that energy into the simple sugars. You know, this is a busy slide, and it's probably not visible to most of you, but that's probably just as well. You won't know again that I'm talking about. But this is one of the things that plants can do as a result of their photosynthetic ability. Not only can they make the major molecules of life that we talk about, the sugars, the proteins, the amino acids, all these things that are basically universally distributed in all organisms and on which we depend for life. And these we call primary metabolites. Primary because they're universally distributed and they're pretty much essential for life. But plants, in addition to that, can make a vast array of what is sometimes kind of pejoratively called secondary metabolites. And that is, these are the chemical compounds that make plants interesting, that make them these chemical virtuosos. Plants have energy to burn, so because of their photosynthetic ability, so there are no constraints on their energy. They can use that energy and they can seemingly waste it. And for a long time it was thought that these secondary plant metabolites, which are things like alkaloids, terpenoids, your essential oils, your tannins, these sorts of things, are all these plant secondary metabolites. And it was thought that they were secondary because it wasn't really understood what their purpose was. They just thought that they were some kind of metabolic waste product, basically, just a physiological garbage that the plant made and sequestered in its different parts. And what has come to be understood probably in the last 10 to 15 years, maybe the last 20 years, is that these so-called secondary metabolites, this vast array of structural diversity that plants have evolved, are not really secondary at all. The purpose that they serve the plant is to mediate the relationship between the plant and the rest of its environment, between the other organisms in its environment, from other plants to microorganisms to fungi to grazing animals and also humans, because we're also organisms in the environment that interact with plants on this molecular level via this molecular language, if you will, that plants have evolved. What this slide illustrates is basically two things. It shows the primary gateway for energy and simple compounds to get into primary metabolism in the plant. That's CO2, water, plus light energy. And that feeds first into the pentose phosphate cycle where you get your simple sugars being created, ribulose diphosphate. And then, these are black boxes, you understand. There's a lot more going on here than is implied. But you get first your simple sugars. That leads eventually down the spine of primary metabolism into your tricarboxylic acid cycle, and thence on into your amino acids, which lead eventually to proteins, and also your lipids come out of the tricarboxylic acid cycle. This is sort of the spine of primary metabolism. What's perhaps more interesting is that at different branches along the spine, you get things coming out that lead into the major classes of secondary metabolites. For example, coming off the pentose phosphate cycle from the simple sugars is a compound called shikinic acid. And that's your first aromatic compound. Aromatic in the sense, not that it smells good, but aromatic in the sense that chemists use, so that it is the precursor to compounds that contain a benzene ring, an aromatic ring. And from shikimate, you get the aromatic amino acids formed, the three essential amino acids, phenylalanine, tyrosine, and tryptophan. And from those, you get a vast array of secondary compounds. The ones that we tend to be most interested in as cyclopharmacologists are the aromatic alkaloids. An alkaloid, by the way, for those of you that don't already know, is a plant secondary compound, if you will, a pejorative term again, but a plant metabolite that's not part of the primary metabolism that contains nitrogen. You can sort of define an alkaloid more easily by what it isn't than what it is. But one of the essential requirements is that alkaloids contain nitrogen. So the aromatic amino acids lead to the aromatic alkaloids, which include things like EMC, psilocybin, multiple hallucinogens, also things like morphine. Many of the drugs of use and abuse that are so economically important are these aromatic alkaloids. In addition to the shikinic acid also gives rise to phenolic compounds such as cinnamic acid, caffeic acid, where these tend to be flavorings and essential oils, flavorings and literally aromatic compounds in the sense of fragrances. This is one of the main branches of primary metabolism that leads to aromatic type compounds. Further down the chain from the Krebs cycle, you get, branching out of the Krebs cycle, you get aliphatic amino acids, things like valine and lysine, that lead to another class of alkaloids, the aliphatic alkaloids, which include things like nicotine. Some of the tropanes are also aliphatic. Pyrrolizidine alkaloids are aliphatic. That is, they don't incorporate an aromatic ring, and they're not derived from one of these three aromatic amino acids that are so important in metabolites. But also coming off of the Krebs cycle is a 2-carbon fragment called acetyl-CoA. And this 2-carbon fragment feeds into a number of secondary product biosynthetic pathways. For one thing, it leads to both fatty acids and lipids, and a class of compounds called the acetogens, which are just many of these acetyl-CoA units joined together, and you get things like eugenol belong to the class of acetogens. But acetyl-CoA can also lead to metalonic acid, and metalonic acid is basically a 5-carbon unit that leads to the terpenoids, and you get the terpenoids are down here. You have the simple terpenes like menthol, which basically are 5 or 10 carbon units. You have the sesquiterpenes. One example is picrotoxin. And the diterpenes, you've heard this morning a lot of talk about salvinorin, and that's a classic example of a diterpene. And even more complex terpenes such as digitonin, and other steroid-related compounds. So these are all basically built up of many units of mevalonic acid joined usually in a head-to-tail fashion. So you have basically four main classes of plant secondary compounds. You have your aromatic shikinic acid derivatives, you've got your aliphatic alkaloids, your acetogenins, and your terpenoids. I mean, there are other classes of plant secondary compounds. Those are the four biggies. This is just an example of some alkaloids derived from these different sources. These are aliphatic alkaloids, and these are the aromatic alkaloids. It's quinidine, which is from the quinine tree, cinchona, an important medicine in treating heart arrhythmias. Tubocurarine from curare, that's used in abdominal surgery and others as a muscle relaxant in certain kinds of surgery. Your beta-carbolines, also aromatic alkaloids derived from tryptophan. Alkaloids are one of the largest classes of secondary compounds and also one of the most structurally diverse classes of secondary compounds. And also, because we get so many important medicines from them, drugs both of use and abuse, and the distinction is really in how they're used. Alkaloids are one of the classes that have the greatest economic importance for us. One of the things that we get from plants that is most important for our health and well-being. This is a slide I haven't put up before, and I'm not sure how much I want to go into it. I don't know exactly how to say this. It's one of the things that's always struck me as curious about the biosynthesis of these aromatic compounds, these things that branch off the shikimate pathway, is what you might call curious parallelisms in biosynthesis. It's either something important or it's so what, ho hum. I'm not sure which. I think it's kind of neat. You know how sometimes people on DOT or sometimes on mushrooms report having the experience that you can kind of turn the circuit board of reality over and see how it's wired, and you realize that, it's not at all what it appears to be. This is sort of on that level. I don't know if that really makes any sense. This particular slide here shows how these three aromatic amino acids arise. This is your CO2 plus H2O plus light. Photosynthesis leads eventually to these two simple sugars, which are formed into shikinic acid and then chorismic acid. And out of that, you get your aromatic amino acids branching off here: phenylalanine, tyrosine, and tryptophan. And then also other important metabolites, folic acids, and so on, which we're not really going to go into. But then if you look at the classes of secondary compounds that come off of each one, there's something that I've always thought was kind of interesting. When you have phenylalanine here and tyrosine, phenylalanine and tyrosine can basically go two ways. You can get decarboxylation to form the amines, beta-phenethylamine coming from phenylalanine and tyramine coming from tyrosine. So basically you've removed an acid group and you've left the amine intact. And then from these simple amines you get things like mescaline, which interestingly enough is the only hallucinogenic phenethylamine in nature, although it's given synthetic chemists such as Sasha Shulgin many ideas for many analogs, but they're not found in nature. You get neurotransmitters such as dopamine, norepinephrine, or epinephrine, and norepinephrine. Ephedrine, another plant compound, that has some of the stimulatory properties of amphetamines. And then even further down the chain, you get complex alkaloids in which these amines, these simple amines, are precursors. They're incorporated into more complex rings, morphine, tribocurate, and so on. The other thing that can happen to these simple aromatic amino acids is instead of losing a carboxyl group, instead of losing an acid group, you can lose the amine. And then you have your phenylacetic acid derivatives. Things like cinnamic acid, caffeic acid. They can lead, these are widespread in coffee and a lot of spices. They contribute to the flavor of coffee, spices, this sort of thing. Things like safrole and nutmeg. Safrole, which is a constituent of nutmeg oil, dill apiol and dill oil, they're also basic simple derivatives of these cinnamic acid derivatives. They are fragrant. Their biological activity is basically that they're volatile. And then these can further join into dimers. They can form dimers and form a class of compounds called lignans, which are things like glutaphyllatoxin. It's an anti-cancer drug. There are many interesting lignans, and they have a whole range of biological activities, ranging from some are antimicrobial, some are antiviral, and some have activity on the central nervous system. And then if they polymerize even further, you get lignins, which are polymers of these C6, C3 units. The C6 in the ring and the 3 carbon units sticking out from it. When it becomes polymerized, you get lignin. And lignin is the major structural constituent of wood. Lignin is what gives trees its strength. And interestingly enough, when I was a graduate student at the University of Hawaii, my professor was interested in stress physiology, and he showed that by growing seedlings under conditions of increased gravity, by growing them, he was interested in gravity as a biochemical determinant, and he showed that he would put seedlings in a centrifuge and germinate them there, and show that the levels of lignans were increased. And not only were they increased, if he did anatomy, if he took sections of these seedlings, he showed that the distribution was corresponded with the points of maximum stress in the plant. So this is, lignification. The lignans are deposited on the cell walls. This is basically a gravity-determined property that governs the distribution of these lignans. I mean, if you make an engineering diagram of a tree where the major points of stress are, that will match exactly where the maximum depositions of lignans are. So, whatever. So that's kind of the story with the aromatic amino acids, phenylalanine and tyrosine, the precursor of the phenethylamines, and the complex alkaloids that come off that. And then this class of compounds, the essential oils and the lignans, and then the lignan itself. Well, if you look at the same thing with respect to tryptophan, it's kind of the opposite. Tryptophan can be decarboxylate or deaminated. If you remove the amine group of tryptophan, what you're left with is indole-acetic acid. Indole-acetic acid, it turns out, is one of the major plant growth hormones. It's the plant hormone that's universally distributed in plants, and it governs the elongation of the plant stem and cell division. So this is basically a very important plant growth hormone. But it doesn't seem to go, there's not a vast array, unlike with the deamination products of phenylalanine and tyrosine. With the indole, there's not a vast array of secondary compounds based on indole-acetic acid. I mean, there are variations. But it hasn't effloresced into a great variety. Where you see the variety in the indole is when you lose the carboxyl group, you get the simple tryptamine, you get tryptamine. And then leading to that, you get your other compounds like 5-methoxy-DMT, dimethyltryptamine, by adding methyl groups. When you add a hydroxyl group in this position, you get 5-hydroxytryptamine, which is a major neurotransmitter, as you all know, also known as serotonin. Again, I don't know if this is neat or ho-hum. It's always struck me as interesting. The dimethyltryptamine is basically 2 steps of the tryptophan. I mean, tryptophan is universally distributed in nature. Here, 2 enzymatic steps away is dimethyltryptamine. The curious thing about dimethyltryptamine, as you all know, is it's not orally active. So it could be widely distributed in a lot of foods that we eat and we never know it unless we happen to fortuitously also eat a plant containing an MAO inhibitor at the same time. Then you know. But you might be happily dining on plants, cereals, for example, high in dimethyltryptamine for a long time. If you've ever combined it with an MAO inhibitor, you wouldn't know. But these tryptamines, there's quite a structural variety within the tryptamines of these simple things. And then even more complex, you get, the next step is beta-carbamines, and then there's a whole vast array of more or less complex indole alkaloids, things like ibogaine, and even more complex, enormous things like vincristine, which is one of the more complex indole alkaloids. This, of course, is an anti-cancer drug. It's used to treat leukemia in children. Rumor has it that it's also psychedelic, or not psychedelic, psychoactive. People undergoing cancer chemotherapy have reported interesting experience with being treated with vincristine. I don't recommend it. It's not something that I would care to try. But another interesting thing, when we saw that in the phenylalanine and tyrosine class, when you get polymerization into the lignans, it becomes the structural component of trees. What happens when the indoles polymerize? You get melanin. And melanin is the black pigment that makes many mushrooms turn black. It's the black pigment when bananas begin to turn black on the outside. That is melanin. And melanin is an indole polymer, basically. The curious thing is that in nature, it doesn't really usually come from indoles at all. It comes from tyrosine and dopamine and these phenethylamine type compounds. So, go figure. Anyway, this just gives you, I guess the point that I'm trying to bring out from all this is that depending on which way it goes, these aromatic amino acids can lead into major secondary product pathways, into large categories of compounds that are basically derived from these things. And if you go decarboxylation, you lead into one category. If you go deamination, removing the nitrogen, you lead into the other category. And there's a kind of a symmetry here. I haven't got it completely figured out, but it's interesting that it exists. This is another example. Sasha Shulgin has talked about the essential amphetamines. Interesting thing. In nature, the only CNS active phenethylamine, the only hallucinogenic phenethylamine is mescaline, which is 3,4,5-trimethoxyphenethylamine. But some of these essential oils, L-amycin from nutmeg, acerone from, of course, calamus, the sweet flag, these sorts of things, if you make it, they themselves are not psychoactive. But if you make a trivial synthetic modification, if you introduce a molecule of ammonia into this at the right position, then you get your essential amphetamines. The simplest, or probably the most obvious one, of which is the 3,4,5-trimethoxyamphetamine. You can think of it as a mescaline analog, which it certainly is, but it's also, it can be made directly from L-amycin by simply adding an ammonia at this position. Similarly, if you take acerone and do the same type of ammonia substitution, you get TMA2, where the essential difference between these two is the position of the methoxy groups, where they are located on the ring. This is 3,4,5, this is 2,4,5. And one of the most interesting discoveries Dr. Shogun made when he was working on this early on, back in the late 50s, was that this simple trivial rearrangement, the moving this one from here to here, from the 3 positions to the 2 positions, resulted in an enhancement of potency of orders of magnitude. Sasha's here. Correct me if I misrepresent any of it. But then he found that by taking various substitutes, so the 2-4-5 configuration was important, and then various substitutes on this 4 position, basically he could hang just about anything he wanted off that and still get some kind of activity. So he started hanging things like halogens off it, or methyl groups. He first came up with BOM, which was known a few years ago as STP, but then perhaps the most potent ones, where this R group becomes things like bromine or iodine, you get DOB and DOI. And these are very potent phenethylamines. They're not quite as potent as LSD, but they're in that magnitude. 1 milligram or less is a above threshold dose. They're also extremely selective for the 5-HT2 receptors, which is this class of receptors, serotonin receptors, that they now believe is intimately involved in the action of these hallucinogens. So it's just interesting that, nature invented compounds like these, and these are widespread in nature, but somehow she never bothered to add an ammonia to these and come up with these. It took a synthetic chemist to come along and do that. I don't know. Maybe nature just never thought of it, or there was some other reason.
Maybe he was part of the
Well, yes, that also is a fact, right? This controversy about synthetic versus natural is really a tempest in a teapot, in my opinion, because as someone said, nature did synthesize the aforementioned synthetic chemists, right? We shouldn't lose sight of that. So anyway, that just illustrates the kind of the variety from the standpoint of the aromatic amino acids, kind of the structural varieties and types of compounds that you get just within that class, just coming off the shikimic acid derivatives. And you could basically say the same thing for any of these other classes, the terpenes or the acetogenins or these other classes. So one of the understanding now is that these are not secondary compounds, that these actually do function in the day-to-day economy of the plant in terms of modifying the plant's relationship to its external environment. There's this biosynthesis for behavior paradigm that we're talking about. And the functions of these plant allelochemicals are defense and or symbiosis or semiosis, in other words, a signaling function, stay away or come closer, or symbiosis. And really these functions overlap. For example, an example of both the semiotic function and the symbiotic function is found in plants' relationship to insects, where the fragrance of the flower and the color of the flower acts as an attractant to the insect. So the plant basically sends out a signal, come closer and become involved, but also it enables the insect to play a role in completing the plant's life cycle. The plant cannot pollinate itself. It needs the insect to help it, at least these particular flowering plants. So this is, it's by virtue of the chemicals that the plant produces that it establishes this symbiotic and semiotic relationship to the insects. This is an example of a defensive chemical. This is Geroia hirsuta that Dr. Schoolteacher has written about quite interestingly. This plant causes, in Peru, a phenomenon called the Supai chakra, the devil's clearing, where this plant grows in primary rainforest for about 20 to 40 feet around it. Nothing grows. Actually, Selaginella grows, but nothing else. And the thinking is that this plant probably secretes a compound into the soil, either from the leaves or from the roots, that inhibits the growth of other plants, inhibits germination. So this is a defensive function. The plant is basically staking out its territory and preventing potential competing plants from moving in on its space. So it's basically saying, stay away, I'm toxic. This plant is chemically unstudied, as far as I know. At least it wasn't for you. Do you know, Dr. Schultz? It's never been examined chemically, but it's in the Rubiaceae, which is a family that's very well known for its biodynamic compounds. So there's potentially some interesting chemistry to be uncovered here. This is another example of a true symbiotic relationship. Sorry for the quality of the slide, but the butterfly would pollinate certain plants, and actually certain types of butterfly that found on its widespread. Probably the best studied example are butterflies of the monarch type, which feed on milkweeds. Milkweeds are full of a class of compounds called cardiac glycosides. They're steroids, so they belong to the class of terpenoids, but they're highly toxic. But the butterfly has evolved enzymatic defenses against it. The butterfly, or actually the caterpillar, is able to feed on this plant with impunity. Not only does it feed on it, but it takes the cardiac toxins into its body and sequesters them in its body. And it itself is not poisoned by them, but when it transforms itself into a butterfly, it puts the plant toxin to use as a chemical defense. It becomes unpalatable to potential predators. And they can see these butterflies, they have learned that, certain butterflies of this class don't taste good because they're full of these cardiac glycosides. So this is an example of an even more baroque, if you will, relationship, chemically mediated relationship between the plants and the animals, where the consumer of the plant has evolved a biochemical defense against the toxin, and then goes one step further and uses the toxin for its own defensive purposes. Well, the tryptamines and beta-carbolines provide a good illustration. There are a class of compounds that we're very interested in here, because we're interested in psychoactive plants, but they also provide a very good illustration of which you might call the parsimony of nature. These compounds tend to have multiple activities depending on what biological targets they interact with. It's not just that they're hallucinogens. They're hallucinogens given that nervous systems are the biological targets they interact with, but they also interact on many other levels. These are the tryptamines up here, the beta-carbolines down here on the bottom. And for example, these beta-carboline alkaloids are, in the presence of UV light, many of them are photoactive. And that's what's shown here. If you take something like 6-deoxyharmalan or harmine, put a little bit on a filter paper disc, and put that on a petri plate which has been covered with yeast or E. coli bacteria, and incubated in the dark, nothing happens. The bacteria grows just fine. But if you expose it to UVA, I believe, or UVB, I forget which, for about 10 minutes and then incubate it in the dark, you get a nice clear zone around the filter paper disc where the bacteria has been killed. It hasn't grown due to the photoactivity of these beta-carbolines. And you can do, reasonably nice structure-activity relationships and that sort of thing. But so in this explains possibly one of the allelochemical functions of beta-carbolines for the plant. In the presence of sunlight, it might help to defend it against bacteria or fungi that might otherwise invade the plant through the leaves or the roots. Another example with the tryptamines. Indole acetic acid, remember we talked about, is a major plant growth hormone. As you can see, it bears a pretty close structural resemblance to our favorite dimethyltryptamine, 5-methoxy-DMT, and dopamine, these cycloactive methylated tryptamines. But it turns out that these, if you treat plants with them, these are found in plants like Desmanthus illinoisensis in the roots. And where they were first discovered and why they were discovered in there is they were looking at plant growth inhibitors. This is a good plant growth inhibitor, and they isolated the fraction responsible and found that it was dimethyltryptamine and other related tryptamines. So at this level, these compounds function as plant growth inhibitors. That is their target. That is their ecological function, probably for the weed, Desmanthus illinoisensis. It probably didn't evolve it so that we could go out and gather it and extract it and make ayahuasca analogs out of it. The same tryptamines in other plants, such as varrolas, so when the organism being interacted with are human, such as these Yanomami Indians, then the target for the tryptamine becomes the central nervous system. And you get the activity that we're familiar with, the hallucinogenic activity. So I guess the point of this is that the compounds can have multiple activities, depending on what molecular or biological target they actually interact with. It's not that they have only one kind of activity. So nature makes good use of these things. They are multi-purpose compounds. And the reason that these allelochemicals have these multiple activities, can target multiple biological, can mediate multiple biological relationships in nature is because of the variety of molecular targets that they interact with. For instance, the photoactivity of the beta-carbolines that we saw with the bacteria is due to the fact that they are able to insert themselves between the base pairs of DNA. And they do that, and then in the presence of UV light, they form covalent cross-links with the base pairs of the DNA, which is basically like throwing a big monkey wrench in DNA. So they pretty well, inactivate the cellular processes at that point. In the absence of light, they don't do this, but in the presence of the right wavelength of light, this can happen. So the beta-carbolines do this, but only if in the right milieu and under the right circumstances. Another class of compounds that do the same thing are also derived from plant. They're called the sorolins. And I don't know if you've heard of Kuva therapy, but it's a kind of treatment for psoriasis where you take sorolin pills for several days. It accumulates in your body and in your skin, and then you go into a tanning salon and you get an all-over tan under UV lights, and it causes the cross-links the DNA in the outer surface of your skin and causes that to slough off. So you come out of it looking all pink and healthy, and then gradually the psoriasis comes back and you have to do it again. It was for a while a promising treatment for psoriasis, a heartbreak of psoriasis. I don't know if it's still being used. And it really is a heartbreak. I mean, it's a serious condition, in advanced cases. Another example of phototoxicity, the example there is the target, in the case of the beta-carbolines, is DNA. It basically throws a monkey wrench into DNA. But cell membranes are another important biological target for these active plant allelochemicals. This is an example. This used to be a red blood cell before it was exposed to a compound called alpha-trithionyl, which is a photoactive sulfur derivative found in marigolds. And in the presence of UV light, alpha-trithionyl sequesters itself in the membrane. It's highly lipophilic. It's highly fat-soluble, so it dissolves in the membrane and literally blows holes in the membrane. So that's what you're seeing here. These craters are literally, the thing is riddled with holes, and of course it's lost its osmotic cones. So another photoactive compound, but a completely different mechanism of action. It works on membranes instead of DNA. Another example of biological targets are enzymes of different kinds. And again, we can illustrate from the beta-carbolines. The beta-carbolines are potent monoamine oxidase inhibitors. They inhibit the enzyme that deaminates neurotransmitters such as 5-hydroxytryptamine. Beta-carbolines block that enzyme. So at that level, they're working on, that becomes the target for them. This, of course, is underlies the pharmacological mechanism behind ayahuasca. Ayahuasca is, I'm sure all of you know, is a combination of two plants, at least two, one of which contains beta-carbolines, the Banisteriopsis, and the other one, the admixture plant, contains dimethyltryptamine. Dimethyltryptamine, taken orally by itself, is not active, because it's inactivated by monoamine oxidase, which is present peripherally in your stomach, in your liver, and so on. But if you take it in the presence of a beta-carboline, the beta-carboline inhibits MAO and protects the DMT from peripheral breakdown, allowing it to be absorbed into the nervous system in the intact state. So this particular action of beta-carbolines, the ability to inhibit monoamine oxidase, becomes important in the human interaction with these two plants that have led to ayahuasca and all the ayahuasca analogs that people are now experimenting with. Another example of an important biological target are second messenger systems. Second messengers are enzymes, usually such as protein kinase and adenylate cyclase. They're enzymes that are usually bound to membranes that mediate what you can call kind of generically post-binding events. They are second messengers. They govern intracellular processes, usually that follow on the binding of a drug or a neurotransmitter to a receptor. And one of the classes of compounds that are one of the most potent inhibitors of second messengers are called formol esters, found in the family Euphorbiaceae. They're very important activators of a protein called protein kinase C. Kinase C is involved in, and one of the reasons that formol esters are highly carcinogenic is because of their action on protein kinase C. Okay, so the point of that is that these plant allelochemicals, these biologically active secondary compounds, have these activities because they interact with some kind of receptor. They have to get in and bind with something in order to do what they do. Now, you can take the term receptor in a sort of broad sense, where a receptor is anything that a drug binds to exert its effect. It might be an enzyme, it might be a membrane carrier molecule, it might be a nucleic acid. All of these are in some sense receptors, in the sense that these biodynamic molecules can bind to them and affect them by doing so. But receptors and their ligands, that's what we call the molecules that bind to the receptors, are a complex chemical communication system that regulates the internal homeostasis of the organism by governing this signaling between cells, systems of cells, and even other organisms. And the speculation is, how did we end up with all these internal receptors? The speculation is that these receptors originally served an ecological function, that they originally were found on the outsides of organisms, and that the biosynthetic diversity we observe in the natural world is the evolutionary consequence of billions of years of this biochemical or chemical ecological warfare. One component is the elaboration of these defensive or aggressive or symbiotic allelochemicals on the part of the plants the organisms that they're targeted to in turn evolve adaptations, adaptive responses to neutralize them, detoxify them, or counterattack in some way. And then the plants respond with biosynthesis of even more novel, even more complex molecules with even more complex modes of biological activity. And as multicellular organisms became more complex, this interchange between organisms, which is basically organisms synthesizing and excreting these compounds into the environment, they travel to other organisms. We're talking about largely one-celled organisms and interacting with receptors on the outside of the cells. But as organisms became more complex and multicellular, these receptor systems gradually became internalized. And the signaling functions, instead of transmitting between organisms, then became adapted to transmitting signals between populations of cells within an organism. So you get hormones and neurotransmitters, which are basically our own ecological chemicals that have been adapted to this internal function. But they were originally there to mediate relations between organisms in an ecosystem. That's how they arose, and then as they became adapted to this internalized function, they became specialized to that kind of activity. This is a typical neurotransmitter receptor, which is sort of more what we think of conventionally when we think of a receptor. This is sort of a narrow definition of a receptor. And this happens to be a serotonin receptor, but any neurotransmitter receptor incorporates much the same processes. In this case, you've got tryptophan, which is the essential amino acid, right off the succinate pathway. And that is transformed within the presynaptic membrane into 5-hydroxytryptamine, or serotonin, as it's also called. That is stored in membrane-bound vesicles until it's used. And when it's used, these vesicles migrate to the membrane of the cell, and the neurotransmitter is released into the synaptic cleft. Then various things happen to it. It travels across to the postsynaptic membrane, and it interacts with one or more different types of serotonin receptors on the postsynaptic membrane. The 5-HT2 receptors, the 5-HT1 receptors are just shown, but there is now, I think there's getting up to be at least 8 different subtypes of serotonin receptors alone. And every neurotransmitter, dopamine, GABA, all these things have their own receptor subtypes, so it's quite a complex picture, really. And then, once they bound to these membrane, these receptors, these specific receptors located on the membrane, it activates second messengers, and subsequent events take place. Once the neurotransmitter has done its work, it's taken back up into the presynaptic membrane through a carrier molecule. This is yet another kind of receptor, and recycled. It's either recycled, repackaged into the membrane-bound vesicles and reused, or at that point it may also be degraded by monoamine oxidase and turned into 5-hydroxyandolysic acid in this case. Now, psychoactive drugs, those that work on the central nervous system, tend to do their job by affecting one or more of these processes that are taking place at the receptor site and in the vicinity of the synapse. They either affect the synthesis, the storage, the release, the receptor binding, or the reuptake, or the degradation of the neurotransmitter. That's how they do their job. For instance, racetine, the alkaloid from the Indian snake root, basically disrupts these vesicles and causes the 5-HT to be released into the cytoplasm and sort of leak out of the cell. So actually it's not selected for only serotonin. It doesn't draw your monoamine type neurotransmitters. But it depletes the presynaptic cell of its neurotransmitters. That's what the sertraline does. Your hallucinogens the so-called true hallucinogens, LSD, psilocybin, these ones, it's now thought interact primarily with the 5-HT2 receptor. In other words, they substitute for serotonin at the 5-HT2 receptor. They also affect some of the 5-HT1 receptors, but that's not quite so well understood. Antidepressant drugs such as Prozac is a very selective blocker of the serotonin reuptake molecule. It gets in here, prevents the serotonin from binding to this and being taken back up into the cell, with the result being that there's a higher amount, a higher availability of serotonin in the synaptic cleft. Prozac is, typical antidepressant in that respect, and a marvel of structure activity engineering because it is so selective for serotonin. So this is this table is undoubtedly hard to read, but this just shows some of the natural products that affect the nervous system. The compound, its chemical class, its pharmacological action, and its therapeutic application. And basically, I think the point of this slide is that you can't think of an activity, a pharmacological activity, for which there is not a natural product that shows that somehow affects those receptors or those mechanisms involved in that activity. And you could put the same kind of slide up for any type of, any class of activity. If you're interested in liver function, we could construct a similar one for the liver. If you're interested in antimicrobial activity, you could construct a similar one for antimicrobial activity. Notice in the case of the central nervous system compounds, how many of these are alkaloids. Alkaloids are disproportionately represented when it comes to the central nervous system activity. It's something about the presence of that nitrogen that gives them the ability to interact with these receptor systems that are so important in the functioning of the nervous system. But any class of activity that you can name as an actual product that displays the activity. This is, physostigmine inhibits cholinesterase. Reserpine depletes CNS monoamines. THC, it says it's psychoactive, but one of the exceptions, it's not an alkaloid. It doesn't have nitrogen. So it's kind of interesting. Nature has evolved these allelochemicals that interact with receptors that we haven't discovered yet, which is kind of interesting. And we've seen a classical example of this in salvinorin. I mean, the work that's been described here is very interesting. A compound has been identified. It has definite psychoactivity, and we at the moment don't have a clue what receptor system it might interact with, except we're pretty sure it's probably not 5-HT2. There are speculation, but that's partly the importance of natural products. Because of the importance of natural products in drug discovery is that this molecular diversity, out of looking at the molecular diversity of natural products, you get new chemical entities come out, and they point the way to new modes of pharmacological activity. I mean, you can make, many phenethylamine derivatives, many tryptamine derivatives, and that's all very well, but you're looking basically, with subtle differences, you're looking basically at a characterized mode of activity. We know pretty much that it involves the 5-HT2 receptors. Some of these new molecules that are coming out of the natural world point the way, may open the door to completely new pharmacologies. Dr. Schoolteacher, a few years ago, wrote an article which has been not widespread at all. It was called Phytochemical Gaps in Our Knowledge of Hallucinogens. And I think it was published in a very obscure journal, the Journal of Plant Science or something like that. And I always thought that it was, of his many very significant papers, I always thought it was one of the most significant, because he pointed out in there, it was a description of about 30 plants for which the ethnobotanical documentation was clear, No doubt that there was probably activity there because people were using it and they were experiencing effects from it in different traditional societies. But there was no information on the chemistry. And I wished that he had called it phytochemical gaps in our knowledge of CNS active plants, because it would have gotten a lot more attention, and it might have induced some of these drug companies to look into these plants more. By calling it phytochemical gaps in our knowledge of hallucinogens, they said, oh wow, who needs another hallucinogen? You know, but as we show with Salvin, Oren, and these sorts of things, you never know what these things may end up being good for. You can't dismiss these. Well, this is another example, just another out-of-focus example of the varieties of CNS activity that you get in the natural world. This is a table that I put together from NAPRALERT. Some of you have heard me talk about NAPRALERT. It's a natural products database created by Norman Farnsworth at the University of Illinois. And among the things that it talks about are different classes of pharmacological activity. I went into NAPRALERT and I asked that if I specify these types of central nervous system activity, analgesic, anesthetic, local in general, anorexic, drug addiction inhibition, learning enhancement, hallucinogenic, I say in NAPRALERT, how many hits are there for each of these types of activity? How many plants are reported to have these different types of activity? So it fits the number facts, right? Analgesic, 3,710 citations for plants with analgesic activity. Hallucinogenic activity, not bad, 88 citations. Anesthetic activity, 226 citations. Tranquilizing effects, 260 citations. And I guess what the point of this is that there are indications of activity far more extensive than have been looked at. The world needs a good, non-addictive, effective analgesic. Chances are you'll find it in the natural world if you look hard enough. The world needs a better tranquilizer. If you went through and you collected all 260 plants that this referred to and started screening, you'd probably come up with a pretty good tranquilizer after a while. So the point is that this information is out there. What's not happening is there needs to be a systematic follow-up to these things. I mean, we talk a lot in the drug discovery game about hit rates. When I was at Shaman Pharmaceuticals, they were always saying, well, what's our hit rate and what's the correlation, to the ethnobotanical data that we're collecting? And I always said, it's a completely ridiculous argument. The hit rate is 100%, right? Because every plant contains allelochemicals that have biological activity. The question is, do they hit a target that's of therapeutic interest to us? That's a more interesting question. I mean, often they don't. Often they hit a known target or something we're not interested in. But the point is, they all have biological activity. You can take any plant at random. If you screen it against enough receptors, sooner or later you're going to get a hit. So the hit rate is 100%. The other thing is that these natural products, these molecular templates that come from the natural world, one of their main things as far as drug discovery is they give synthetic chemists like Sasha ideas. And synthetic chemists, Sasha accepted, tend to be kind of an unimaginative group. They really can't come up with very many ideas, but they can look at the natural compounds and they can say, oh, well, we'll tweak it here and tweak it there and maybe modify its activity, come up with something a little better, with a better selectivity for a given receptor, maybe less toxic, or maybe longer lasting, or something like that. And that's what's going on here. This is just an example. And you have the natural compound here, morphine, which is a non-selective opiate agonist. Very good stuff, very important in medicine, right? Important analgesic. But then the synthetic chemical comes along and turns it into now Trindol, or that is the looks at the structure of morphine and comes up with an idea for modifying it. So it incorporates the basic morphine into this. And this becomes a delta opiate selective antagonist. So while morphine is a non-selective opiate agonist, hits all the opiate receptors more or less. This is a blocker of one of the opiate receptor subclasses. Very important to be able to introduce that selectivity in the molecule. Another example, cocaine, a central stimulant, a local anesthetic, being used to label the dopamine uptake carrier because it binds very selectively to that important drug, again, of use and abuse. The synthetic chemists come up with CFT. It's an even better label of the dopamine uptake site than cocaine itself, and it's 3 to 10 times more potent than cocaine as a psychomotor stimulant. Just what the world needs. Ibotenic acid from a mushroom is an NMDA agonist. The NMDA is N-methyl-D-aspartate, one of the cytosine amino acid receptors, and this is an agonist of that receptor. So it has, among other things, anesthesia, potentiation, and it inhibits tremor, and it inhibits emesis. But synthetic chemists come up with AMPA, which is a structural analog of this, and this becomes a potent glutamate crystallizing agonist. So basically, the take-home lesson here is that by making relatively trivial modifications in natural compounds, synthetic chemists can tweak their activity. They're able to selective for certain substrates, or even turn agonists into antagonists, so they have the opposite effects. And this is part of the game, again, of coming up with new drugs, new therapeutic compounds, but natural products lead the way because they give the chemists the ideas for things to work with. Well, on a more intimate level, you could say that humans really value plants in some sense. If you want to be reductionist about it, humans value plants for the chemicals that they produce, which we find useful for some reason, because we either put them into our bodies or we put them on our bodies, and we find them useful. Spices, fragrances, cosmetics, drugs, and, of course, food also has to be included in this category. We depend on the biochemical diversity of plants and the biochemical versatility to give us these products which we find indispensable in our economy, in our health maintenance, and in our lifestyle. For example, plants contain flavorful compounds, these terpenoids and polyketides that give spices, such as nutmeg and ginger, their flavor. Drugs, as we've said, opium poppy is probably the primary example. And in other cases, they yield pigments, they yield dyes, which we use to dye our clothes or put in cosmetics to put on our own body. Well, I've always thought that one useful way to look at the array of these biological activities and the array of allelochemicals when it comes to thinking about human interaction with plant secondary compounds. In other words, I'm talking not so much about other organisms, I'm talking specifically about the human relationship with plants and how that's chemically mediated via this molecular language that plants speak. And a concept that seems to me to be useful is you can class all these things as sensorotropic chemicals. I don't know if you'll find the term sensorotropic in any dictionary. Probably not, because I coined it. But what I mean there is they are all these allelochemicals from plants impact on our senses in some way. Sensorotropic chemicals are chemical messengers which are produced by plants which transmit their message by interacting with sensory functions in some manner. And this includes pigments, aromatic compounds, spices, flavoring agents, and pharmacologically active compounds such as drugs. And one useful way to think about these sensotropic chemicals is to picture them arranged along a spectrum. I don't have a slide, but if you, over on this side, you have your sensotropic chemicals that are utilized primarily as food. In this case, their impact is primarily on the taste receptors and to some degree on the olfactory receptors, and also visual. I mean, that's the level on which we interact with food. If it looks good, smells good, and tastes good, it's food. But what gives it those properties is largely the plant's secondary compounds that's in the food. Toward the middle of the spectrum, you get compounds which, you get things that sort of, they're definitely foods, things like chocolate, which is shown here, one of my and Jonathan Ott's favorite sensotropic chemicals, are the flavoring agent that gives chocolate its unique qualities. Well, but chocolate also contains alkaloids, which are drugs. Is chocolate a food or a drug? I don't really know. It's actually both. It's a moot point. The point is they fade into each other. You can't really separate foods on this side and drug and flavors somewhere in the middle and drugs over there. But there is a tendency along the spectrum for pharmacological activity to become more prominent as you get into the more drug-like things. For example, herbs. You go into any herb stall in a third world country, this happens to be in Florencia, Colombia, and there will be any variety of aromatic plants that you can buy from the herb lady, and they'll say, put it in your bath, it's good for this, it's good for that, put it in your bath or make a tea and rub it on your body, and if you want, you can also put it in your salad. It's definitely a food, but it's also a drug. These herbs are drugs as well as are their medicines. So where is the distinction? And then finally, on the far side of the spectrum, you get an even greater elaboration. You get plants that are more unambiguously drug-like. They tend to be more toxic. Their active principles tend to target specific receptors. They tend to be a higher molecular weight than the aromatic compounds you find in herbs. And, they're medicinal plants, definitely, but they're still on this spectrum of sensotropic plants. So, on this spectrum, the foods and the aromatic and fragrant plants, the flavoring plants and the spices and the drugs all sort of really fade into each other. And so, it's not really a valid question to say these things are drugs or foods or flavorings. They're really both, and they're all sensotropic chemicals. This is supposed to be opium visions. I don't know if you can see it. This is the Chinese fellow. But another aspect of the use of these pharmacologically active plants, when humans, this is different folk remedies in the marketplace of the Quitos. You can go into the marketplace and they have these bags of dried plant material set out with their common names and what each one is for. And you can just buy your medicines. You don't go to the pharmacy, particularly if you're poor, if you're in that economy, you just go to the local herbalist and buy the medicines that you need. And what this is illustrative of, in a way, is that when it comes to the human interaction with plant allelochemicals, we take the plant chemicals, which the plants evolved as defenses or symbiotic chemicals or fragrances or whatever for its own purposes, and we put those chemicals to our purposes. We make a deliberate implementation of an allelochemical interaction to achieve some desired therapeutic effect. Now, the plant didn't evolve these things so that we could use them. We happen to find these chemicals useful for our purposes. So there's an element here of human intervention, of human adaptation of an allelochemical to serve a purpose that may be very different from the reason that the plant originally evolved that particular compound. But this can have profound influences both on the humans that utilize the plant and on the plants themselves. For example, this is Dioscorea. which is the source of complex plant steroids. And in the plant, the steroid compounds that are found in this probably serve a defensive function, because they can interfere with insect molting, and they can interfere with the development of the insect life cycle, and essentially protect the plant against being decimated by insects by interfering in the molting process. The insect eats the steroid, and that basically throws a whole monkey wrench in its life cycle. However, humans come along and take the same plant and extract the steroids out of it and turn it into an effective oral contraceptive. This is far removed from the ecological function it served in protecting the plant, but the human use of the steroid has greater evolutionary consequences for the plant, greater long-term consequences, because it leads us to domesticate the Dioscorea, to cultivate it, to install plantations of it so that we can get this valuable chemical out of it. And that affects the future evolution of the plant. What's happened to the plant is it's become domesticated, and that is going to affect it for as long as the species exists, as long as we still find it useful. The development of these agricultural techniques, which was kind of an enhancement of this allelochemically mediated interaction or relationship between humans and plants, led us to evolve from a nomadic foraging lifestyle to a sedentary lifestyle. And cultivated plants instead of primary plants became the source of our basic necessities. And this rooting of formerly nomadic populations to permanently inhabited sites where crop cultivation took place season after season and year after year led first to the establishment of villages, eventually city-states with larger populations, division of labor, complex political organizations, systems of law, science, technology, based largely on the storage and transmission of written information. But it was the invention of agriculture, the first real formalization of the plant-human symbiosis, which defined a relationship between humans as plants and plants as something qualitatively different from simple foraging of herbivores on plant populations. It was this formalization of the relationship, the creation of domesticated plants, that made possible the milieu in which civilization arose and could flourish. And if this process had a profound impact on human culture, it also had an equally profound impact on the food and medicinal plants, which were the basis of this agricultural revolution. This establishment of sedentary agriculture set the stage for the process of plant domestication and selective breeding. Initially, this was an empirical process, an empirical human activity based largely on observation and trial and error. Farmers would unwittingly select for those specimens which displayed an enhancement of some desirable property: a sweeter or larger fruit, an unusual pigmentation, a pungent fragrance, a less bitter taste, a more bitter taste, indicating high levels of alkaloids. Who knows? Basically, this process is a process of modifying the chemical properties of the plant to suit our purposes, to increase the biosynthesis of those secondary compounds we consider important, or to lessen them, to get rid of the bitter ones, and that sort of thing. So this led to the development of cultivars. Plant cultivars are basically genetic monstrosities, such as shown here. They don't occur in nature. They couldn't survive in nature. They're entirely dependent on human propagation in order to exist. And most of our major food plants are cultivars, and most of our major medicinal plants as well. Cannabis sativa is a good example. It probably doesn't even exist in the wild state. There are many different strains, many different cultigens that we have bred over thousands of years for different purposes: oil, fiber, and resin. But the cannabis is a good illustration of a plant that is basically almost entirely exists as a cultivar now. Salvia divinorum may be a similar example. Tobacco. There's another one. Actually, that wasn't tobacco. That was belladonna. In fact, one ethnobiologist even suggested that the pharmacological utilization of plants could not appear as a specialized branch of human knowledge until the ongoing process of plant domestication had led to the development of cultivars with reduced levels of allelochemicals compared to the wild types. And this happened as centers of civilization grew up and people began living in more concentrated areas. You had sanitation problems and transmission of disease problems. His theory was that as long as you were eating wild plants with low levels or with you had a certain constant level of allelochemicals in your system, this was protecting you from infection. When these people started eating domesticated plants, they became more susceptible to disease. Their immune systems were not as stimulated by these allelochemicals. So it then became necessary to go out and gather medicinal plants and reintroduce them into the diet, because the cultivated plants were no longer providing the protection that they required. Now we hear about people... Yeah. Timothy Johns. Do you know his work? He wrote a book called With Bitter Herbs They Shall Eat It, which is a very interesting, which is a quote from the Bible, some part, a very interesting study of the importance of allelochemicals and the way that allelochemicals have mediated plant domestication. He started out doing his work on potato cultivars, believe it or not, in the Andes, and then from that, looking at how these less toxic varieties of potato became domesticated and integrated into cultivation, came up with these ideas. Very interesting book. Well, now they're talking about using plants to do, to carry this one step further, not just breeding plants to modify their chemical properties, but actually breeding through molecular engineering, through genetic engineering. They're talking about breeding plants to produce things like plastics within the plant. And, I've even heard it said that maybe we can get them to produce manufactured products. So instead of buying your VCR in the store, you just go pluck it off the tree. We're a bit far from that, but this is in principle just a further extension. I don't see anything unnatural about it. If we can get the plant to do it, okay. We and the plant, it's a reciprocal give and take relationship. Well, I think the argument, now I'm sort of speculating, moving more into the area of speculation. I think the notion that humans have influenced the evolution of plants profoundly, we can all sort of accept that, because clearly we've domesticated plants, and in the process of domesticating them, we've modified their forms and their chemistry. And we've modified them in ways that are desirable to us, that enhances their usefulness or appeal to us in some way. The other side of the coin is not so clear, and this is the part that I want to kind of speculate on, but we know that we have influenced the plants. The question is, how much have the plants influenced us in our evolution? And is there any evidence that they have been? Certainly in the iconography of artists, we get suggestions that the feeling that there is a more intimate association between plants and humans that we might like to acknowledge. This is a plant man. I'm not sure if it's a painting.
Archibolda.
Yeah?
Archibolda.
Archibolda. This is a mandrake. Sorry, it's the mushroom shaman of the solid plain. We've all seen this, I'm sure, many times. What these images basically suggest is, maybe humans and plants are not that far apart. Maybe there is an even more intimate symbiosis than we realize. And could this symbiosis, and perhaps even the evolution of humans into what we are, have been influenced somehow by plant allelochemicals in some way. The interesting thing is that humans and the precursors to humans have been interacting with these plant allelochemicals. They've been chewing them, snorting them, smoking them, brewing them, and otherwise ingesting them for 65 million years or so, give or take a few, which is basically when the angiosperms began to flower and became really widespread. Not that the gymnosperms and seedless plants don't have secondary chemicals, but the angiosperms really carried it to sort of its level of complexity that we see now. So is it possible that this long association between mammals, between herbivores, and physiologically active allelochemicals might have evolutionary consequences for primates and us? I mean, we evolved from the primates. Certainly this is well known in insects. It's known that chemically mediated coevolution between plants and insects profoundly affected the chemistry and physiology of both. So similar processes of biochemical adaptation may have gone on between plants and plant-consuming mammals, including primates. The evolution of complex secondary compounds, particularly alkaloids, in the angiosperms, and the concomitant evolution of mammals may have been related. It seems likely that early in the evolution of mammals, tens of millions of years before anything even resembling humans appeared in the evolutionary scene, the process of biochemical and genetic adaptations to plant toxins was working subtle changes in mammalian physiology, creating selection pressure for toxin resistance, biochemical adaptations involving the evolution of multiple substrate, highly efficient inducible enzyme systems, such as the amine oxidases and the cytochrome P450 microsomal enzyme systems. These are essentially mammalian adaptations to metabolizing these plant allelochemicals, to neutralizing these allelochemicals. This is part of the biochemical warfare that we were talking about. Constant low levels of these allelotropic and psychotropic plant chemicals in the diet of foraging primates or early humans could certainly have affected their phenotypic biochemical adaptations. They could also have affected their immune, genetic, and sensory and cognitive functions. For example, many plant toxins are mutagens, and they can impact on the inheritance process directly if they should cause mutations in the germ cells, in the egg or the sperm. Unfortunately, as far as we understand evolution, the majority of mutations are detrimental. But from these, some are favored by selection and result in evolutionary adaptations, and those flourish. So it's possible that over many generations, a constant exposure to low levels of mutagens in the diet might have contributed to enhanced mutation rates or enhanced genetic drift of a population, so that perhaps over time the mutation rate was actually accelerated, and so the overall rate of evolution was accelerated. Another area where plant allelochemicals could have had an effect is in the modulation of our immune systems. These dietary allelochemicals could have directly affected the immune response of primates. There's been some recent work published in Germany in which the effects of extremely small doses of toxic alkaloids and other allelochemicals on immune functions was investigated. These were 100 to 1,000 times less than what we consider the pharmacologically active dose. Much less than any dose thought to cause any toxicity or really any kind of response at all. But what these people found was that at these vanishingly small doses, 10 to the minus 12 molar and lower, right? So down in the picomolar and femtomolar ranges, many of these compounds caused a very strong activation of the nonspecific immune system. When these doses were increased to putatively therapeutic doses, the level of cytotoxicity or whatever, they became immune suppressants. They became strong immune suppressants. So these investigators, their interest was to explain the mechanism of action of homeopathic medicines. And they felt that this perhaps explained it, that these extremely dilute homeopathic medicines working at at 10 to the minus 12 molar and lower concentrations cause the stimulation of the nonspecific immune system. But the relevance of this finding for the present argument is that these chronic low levels of these dietary allelochemicals could have functioned to potentiate the immune functions of certain individuals, thus conferring a greater resistance to disease, longer life, enhanced reproductive potential, and all the evolutionary consequences, all the evolutionary advantages for those that foraged on these immunostimulating allelochemicals. Again, it's probably hard to test this hypothesis, but it's a reasonable notion. One of the puzzles of evolution, certainly, in the evolution of primates, is the explosive evolution of the human neural hardware. The size of the human brain tripled in a mere 3 million years, during which time we went from languageless, tool-less, four-legged primates to two-legged, tool-wielding, verbally expressive, and deity-worshipping human beings. All this happened while the invention of writing was still 100,000 years in the future. Of course, everyone knows that the fossil record is full of gaps, but it still is the most tangible physical evidence we have that something very peculiar was going on in hominid evolution since about 3 million years ago. That date, more or less, give or take 500,000 years, marks the appearance of Homo habilis, the first unmistakably human primate. The first proto-human probably walked upright, may have used simple tools, and displays a dramatic increase in cranial capacity and brain size relative to his close cousin, Australopithecus bossii. The most remarkable fossil, this one here, ER 1470, has been dated somewhere between 2 and 2.8 million years. ER 1470 has a cranial capacity of 780 cubic centimeters, nearly twice that of the contemporary Australopithecus africanus, which is shown here, and even larger than other Homo habilis species. This guy essentially is like us, but he lived 3 million years ago. So it shows that something evolutionary pressure was going on about that time out on the Serengeti plain in Africa, where all this stuff was taking place. What caused this explosive expansion of the human neural capacity, of the complexity of the human brain? Well, one of the anomalies of our existence is that we are very much unlike any other species that inhabits this planet. We're the only species with a complex language. We're the only technology-utilizing species. We're the only species able to store accumulated knowledge in a symbolic form external to ourselves. We're the only species for whom symbols, which are ideas of abstractions, have as great a reality as physical objects. This preoccupation with symbols, with abstractions, is the cause and the result of human culture. And it is culture, this abstract edifice composed of symbols, composed of art, science, medicine, artifice, myth, folklore. All of these things are symbolic structures that truly sets us apart from biological species. So, yes, we're part of nature, but we're also quite anomalous. Our species is unique in having these symbolic functions. At the same time, it's clear that much of this symbolic underpinning that carries our civilization forward and that makes us what we are is derived from, or perhaps the inevitable result of, the extraordinary complexity of the human brain, the complexity of our nervous system. Our neuro-linguistic portions of the brain, combined with our associative capacities, our acute eyesight, our tactile sensitivity, and fine motor control, all of which are reflected on the neural level by complex systems of organization that govern all these functions. These essentially give us the neural hardware, the wetware, if you will, that is needed to run the software of cultural programs, right? The symbolic capacity. So, really sticking my neck out here, I want to suggest that perhaps one explanation for this is that some of these psychotropic allelochemicals, specifically some of these hallucinogens, which were probably widespread in the environment that these primates evolved in the forms of mushrooms like Psilocybe cubensis, may have directly or indirectly affected the evolutionary development of human neurolinguistic adaptations. Our large size and complex neural organization of the brain is directly related to our linguistic and symbolic abilities. The neurons that control the lips, the tongue, the facial muscles involved in speech are disproportionately hypertrophied in the human brain. The brain also has large specialized internal structures devoted to language and understanding and processing language. Well, if you think about what language is, and it's really language that distinguishes, language is that, both written and spoken is the vehicle that on which we erect these symbolic structures. A fundamental characteristic of linguistic and symbolic systems is the intimate association of sounds and images. Language is literally imagination in the truest sense. Have an agreed upon meaning. I can say table and you think of a table, or you visualize a table in your head. So you have I make certain sounds and that triggers certain pictures in your imagination. That's why you can understand me. Reading written words is another extension of this. My eyes see the words, I can hear the words in my imagination, and that interior speech evokes the same pictures that speech evokes, if heard. Well, it seems to me that what underlies this process of linguistic understanding, that what mediates this symbolic process, is in a way a process of synesthesia. Synesthesia is experiencing one sensory modality in terms of another. It's like you can see sounds or hear colors. Well, hallucinogens are one of the few chemicals, one of the few drugs we know of that can reliably induce a process of synesthesia. It's very commonly experienced under the influence of hallucinogens. It's one of the few instances, except that we routinely engage in this process of language, which is a synesthetic process, but we don't think about it very much. But then when we take a hallucinogen and experience synesthesia, it seems remarkable. We say, well, I heard I saw the sounds, or I heard the colors, or whatever, and this seems remarkable to us. But there may have been a time in human evolution when this association between the spoken word or meaningless sounds and meaningful images was not so obvious. And could it have been a synesthesia induced by a plant hallucinogen which provided this triggering event that led to the realization of the equivalence of sound and image, and hence gave rise to the first human language? This is clearly speculation, but this is one possibility where these plant hallucinogens may have played a role in human evolution and into the evolution of the language and symbol-using species that we are. Obviously, it's hard to check this. Nobody can prove me wrong, but I can't prove myself right either. So at that point, I think we have to leave it there. This is a painting by Pablo Angelico, the Peruvian painter, that shows in his paintings he represents synesthesia. Synesthesia is a common experience under the influence of ayahuasca, and he represents it by these wavy lines that surround everything. This is how he shows synesthesia in his paintings. So that's quite enough, I think, and that's all I was going to say. So, sorry. So long-winded. Do you have any questions? Good.
I guess probably everybody has, or maybe not, but everybody has heard of some evolutionary example that seems to not quite fit into what we normally think of as evolution. I like the example of the hammer orchid and the wasp, which is required to pollinate it. The hammer orchid smells like the female wasp. The male wasps are hatched out or a week earlier than the female wasps, and the hammer orchid flowers at the same time that the male wasps are coming out. So there's lots of male wasps around and no female wasps, so they all happily pollinate the hammer orchids. Mm-hmm. And the evolutionary idea that there was a trial and error narrowing in on this particular scent, such that now we got it and now it works. But in that case, a miss is as good as a mile. Mm-hmm. And it seems like many of the examples here also, it doesn't seem like you would hunt around in this absolute myriad molecules, universe of molecules, by trial and error and find out the right one.
So in other words, how could it have taken place through random selection?
Yeah.
Right.
And you can postulate all kinds of things like non-local mind and what have you.
Right, or morphogenetic fields or whatever. I mean, it's hard to rationalize some of this. I think it's clear that, natural selection is one of the processes that operates in evolution, but clearly not the only process. I think they're now beginning to realize, well, we were speaking, an artificial life specialist says, there are other boundary conditions on this process that put constraint on natural selection. That's only one of the things. There are other determinants that which are not fully understood yet, which I think must explain why these things seem sometimes more directed. It's not exactly design, but it's there's more to it than just random throwing up of molecular templates and selecting out the one which seems to fit it. I don't know. I mean, yeah, you might have. Yeah.
Have you talked with them or Anglos at all about the notion of external chemical agents having incorporated into multicellular organisms later on
now being adapted to internal functions? I have not talked with her about it, but I think it's a notion she would be sympathetic to.
Yeah, it's scaled down from central organisms being incorporated into each other.
Right. And there's been interesting work, if you look at, there's been work looking at different receptors. Certain bacteria, for example, have endorphin receptors on the surface of their outside. Well, what does endorphins do for bacteria? Who knows? But it's not what they, that was probably the original function of endorphins. And then we, or complex organisms, they became adapted to internal function. And it's true for just about all our neurotransmitters. And, you can find these things in microorganisms as well. So, yeah.
Now, we were looking at the synaptic cleft and something like serotonin being released and then getting a receptor.
Mm-hmm.
It must take a certain amount of transit time and a certain amount of time back again, causing a certain amount of cycle time. These psychoactive hallucinogenic drugs, do they have a different transit time, and is this what causes the slight variation that turns into hallucination states, or what really happens to them?
What really happens in the cleft? You asked the $64,000 question. It may be variations in transit time. It may be another factor is that the exogenously introduced hallucinogen tends to, essentially massively flood the system with an excess of these chemicals, whereas internally they're released in small amounts in specific places in specific times. It may be that, it's more when they're exogenously introduced and all your receptors are flooded immediately with something like dimethyltryptamine, for example. You get the acute, overwhelming hallucinogenic response. Well, dimethyltryptamine is in the brain, itself, but it may only be released in small amounts under certain, at certain specific sites. So you never get an internal effect like a DMT flash, because it doesn't work that way.
Probably also a problem of permeability.
It's also a
because
problem.
that brain barrier is always in between you and.
That's right. But many of these things readily cross the blood. And transit time is one of the things. The other thing is maybe they don't, maybe they fit into the receptor in a different way, and they don't evoke quite the same type of response. I don't think it's well understood why they're different, Yeah.
Sometimes you have artists, drummers, I'm thinking of particularly, who report synesthesia as a result of artistic activities.
Mm-hmm.
So you would think that there is an endogenous mechanism.
There is an endogenous mechanism, definitely. In fact, your comment, I recently remember seeing an article about a woman, a doctor in England, who was studying a person who, what was it? She was able to reliably see colors, or rather see sounds, that he could give certain tones, and she had a genetic, it was two sisters. That was the thing that was interesting. They both had this ability. They weren't identical twins, but I believe they were twins. And they, they both had very specific synesthetic responses to certain tones, but they weren't the same, right? Person A saw blue at the key of C, and the other one saw yellow or something, but they could both consistently see it. So they're beginning to find out there is definitely an internal mechanism for synesthesia, at least in some people. And, as I say, when we engage in a symbolic activity, we do it routinely. We don't even think about it, but that's really what's happening. So, not that I'm aware of. Yeah.
Dennis, I think I asked you about this a little bit before, but it's clear that some psychoactive chemicals are actually defensive chemicals. They're there to slow down and stop herbivores. For instance, penicillin is found in the spineless cactus. A lot of San Pedro has relatively loose spines compared to other cactus. It looks like the plant's investing in defensive chemicals rather than spine types. Maybe LSD and morning glory is there to slow down mice. So what puzzles me is that since DMT is so widespread in the plant world, and maybe it's serving these other functions that you mentioned in the plant world, why don't plants more often just mix in some beta-carbolines and have an actual defensive chemical?
As a defensive chemical, in addition, well, I guess I don't know. The short answer is I don't know. Right? They do. They're very close biosynthetically, and you often get plants that have both. But usually, like one or the other usually predominates. Like in Varrola, for example, you get primarily tryptamines and only traces of beta-carbolines. In certain malpighiaceous plants, such as Banisteriopsis argentea, or now a different species, I understand, but you get primarily, which one is it? Muricata. That's right. You get primarily beta-carbolines, but also traces of DMT. Why it doesn't happen more often, I don't know. It may be that what these tryptamines generally, the function they generally subserve in the plant is more of a plant growth inhibitor than being targeted at herbivores. And in that case, there's no need for the MAO inhibitor. If they're biosynthesized as a defense against herbivores, then you're right. It would make sense to have beta-carbolines to render them orally active.
Also, it's possible that it's just, in the case of the herbivores, it could simply be the bitterness that's involved.
It's also
as
right.
a defensive function, not the psychoactivity.
Right, right.
Although we tend to see it in terms of
But we can, again, that sets us apart in terms of our behavior, and we can perceive a chemical quality of a plant, such as bitterness, which would drive, any sensible herbivore away. It wouldn't want to eat it. And we say, oh, great, it's bitter, it must make a good drug. So, then we
it's known
go for it even more.
It's known, for example, in the case of secondary compounds in nectars, that different classes of animals that are pollinating species, simply you have birds, bats, which are mammals, and then insects, react differently to different categories of secondary compounds. And mammals seem to be vastly deterred by alkaloids in nectars, but bees aren't. And so it's another subtle interaction.
Right. Again, probably because the bees, they probably aren't bitter. I mean, they probably have a whole different set of receptors and probably perceive these things differently. This is the result of this biochemical coevolution, evolution of these receptors, and how the animals that interact with plants perceive these chemical qualities.
Some of the simple toilet organisms like mannatones also have MAO, or the DMT affect them more strongly?
They certainly have MAO. You mean, would DMT be orally active, for instance, in them?
Some of the similar cellular organisms. A lot of times it's found in boot barn, so it makes you wonder if
Right.
soil organisms.
It's very possible. One speculation, for instance, psilocybin is, serotonin affects the serotonin system. Snails and those sorts of things love to feast on mushrooms. So perhaps what psilocybin really is a snail poison, a snail defense. I mean, it certainly is, might protect the mushroom from being consumed by snails in that respect. But then, since it's valuable to us, or since we're interested in it, it affects that whole relationship as well. So I don't know. I mean, it's speculation, a lot of this, but good enough. Thanks for bearing with me. Sorry it was so long.